This is lecture 6A, part 2. The lungs, as I said, the left lung has 2 lobes and the right lung has 3 lobes. In the left lung you have a cavity or space which permits the heart to be in that position. That's called the cardiac notch. What you have is the vascular and bronchial connections going into the lungs. On the left and the right side, they go in at the root. The top of the lungs is called the apex, and the bottom is the base of the lungs. When you look at the thoracic cavity, you can see that it's actually divided into 3 areas. You have the right lung, and you have the left lung, and they are surrounded by serous membrane on the surface. That is called the pleural membrane. It's the visceral pleural membrane that covers the surface of the lungs on both sides. And then you have the parietal pleura, or parietal pleural membrane, which covers the wall of the thoracic cavity on each side. They're completely separate from each other. So you have a visceral pleura on the organ and a parietal pleura on the wall of the cavity. The space in between the 2 pleural membranes is called the pleural cavity, and it is filled with a fluid called a serosal fluid. These are, in general, these membranes are called serosal membranes. We'll see that a bit later. But you can see that the right bronchus and the left bronchus, right primary bronchus and left primary bronchus, as well as the right pulmonary artery, left pulmonary artery, and also veins, all go into the lungs at one area, which is the root, and on the other side as well. The heart itself is within the pericardial cavity and has pericardial membranes, and you have a visceral pericardium and on the organ itself, and a parietal pericardium outside of that. So, but we're talking just about the lungs, so it's important to realize that there are these membranes and there's a cavity in between filled with fluid. Again, this is the figure showing the tremendous bifurcation or splitting of the transport zone bronchi and bronchioles. Now, it's important to understand that in terms of blood supply to the lungs receive 2 separate circulations. One is the pulmonary and the other is the bronchial circulation. So this is arterial blood supply. You have pulmonary arteries, which carry systemic blood from the right ventricle up to the right and left lungs, and that's blood being carried up there to be oxygenated. So we're talking about the pulmonary circulation. This supply of arteries carrying blood away from the right ventricle of the heart is feeding that less than fully oxygenated venous blood up to the lungs to get to be fully saturated with oxygen. The pulmonary veins carry blood away from the lungs back to the heart, back to the left atrium. What you should understand is that you have a second circulation, which is the bronchial circulation. Here, these are bronchial arteries that are branch off of the aorta and carry fully oxygenated blood to lung tissue that can't receive oxygen from any air in the lungs. The only lung tissue that can actually receive oxygen directly, just because it's close to air, is the wall of the alveoli. The rest of the tissue has to have its own blood supply from the systemic circulation, and that is supplied by the bronchial arteries. Bronchial veins, which are returning that blood back to the heart, will connect or anastomose with pulmonary veins and carry that blood back to the left atrium, actually. So there is a small amount of return of less than fully oxygenated blood within the fully oxygenated blood that's going back to the heart through the pulmonary veins. You have a small amount of less than fully oxygenated blood coming back through the bronchial veins. So those are the two blood supplies, the pulmonary circulation and the bronchial circulation. Again, these are the pleura or pleural membranes. These are serosal type of membranes that every cavity, in fact, in the body is surrounded by these two types of membranes. There's a parietal serosal membrane on the wall of the cavity and a visceral serosal membrane on the organs within the cavity. That's true for the abdominal cavity and the thoracic cavity and the pericardial cavity. These pleura are thin double-layered membranes. There's always a parietal pleura, as I said, that in this case, parietal pleura, it's called, this type of serosal membrane is the parietal pleura, and it covers the wall of the cavity, and the visceral pleura covers the surface of the lungs themselves. The major functions, as I told you, I remind you, are these four: pulmonary ventilation, external respiration, transport, and internal respiration. So when you look at, through evolution, at mechanisms that have developed for gas exchange, you see that there is different kinds of increasing levels of complexity in different kinds of respiratory adaptations for gas exchange. Some of the common features, however, for all of these systems in larger animals is the fact that they all contribute to the maximization of gas diffusion rate. So all the modifications occurring or adaptations occurring through evolution increase gas diffusion rates by making modifications that allow for more gas diffusion. And that includes an increase in respiratory surface area, so you have more surface area for exchange, an increase in the partial pressure gradient across the respiratory surfaces by making them thinner, decreasing the thickness, so that helps increase the partial pressure gradient. Ventilation of the outer surface with air and perfusion of the inner surface with blood. And here you see these kind of different systems in different animals. In amphibia, some amphibia, you have the exchange structures actually external, hanging out in this manner here. You see them right here in this area here on either side of this salamander. In some invertebrates, you have actually pores that lead into a space where you have internal gills that can exchange oxygen and CO2 between the water and the blood, so that oxygen goes into the blood cells and CO2 goes out. Some insects have these holes or spicules throughout their body, leading to channels that permeate their whole body. And then, of course, mammals have lungs and trachea, fully internalized, specialized tissue for gas exchange. Here you see a diagram showing the structure in an insect. These spiracles are holes along the side wall of the organism that distribute air throughout the body. In fish, you have maximization of gas exchange by developing a large surface area, and that is the gills. The surface is constantly ventilated or perfused by water, actually, in a unidirectional manner. The water goes through the mouth of the fish. This is supposed to be a fish. It goes through the mouth of a fish. Let me see, how can I draw a fish? There you go, there's your fish. That's not a propeller, those are the back fins. Let's give it a little eye. And most importantly, the gill covers, which are here. And so water is going through into the mouth and then out through the gills and gill covers on either side. The gills are structures that are highly vascular, bringing blood very close to the surface so you can get gas exchange with the water, from blood to water and water to blood. This is a countercurrent kind of apparatus in that, and I'll show you in the next few slides, but that's a very important concept, countercurrent flow. Well, the countercurrent mechanism of gas exchange that's occurring is that the water is flowing in through the mouth and over the gill structures that contain the blood vessels, and then out through the gill covers. Meanwhile, the blood is flowing in the opposite direction. This allows for optimal exchange, the optimal concentration gradient in terms of concentration of oxygen between the blood and the water. There's always a difference. There's always a good concentration gradient. If they both flowed in the same direction, then there would only be a good concentration gradient at the beginning between oxygenated water and poorly oxygenated blood. But by the end of that pathway, you would have water that had less oxygen, and you would have blood that had more oxygen, and so the difference, the concentration gradient, wouldn't be as great. In this way, with a countercurrent flow mechanism, you get optimal concentration gradients over the entire length of the path where the exchange occurs. It's a concept that comes up often actually in biological systems, and we'll talk about it again when we talk about renal physiology. And it's also a mechanism that is used for conserving heat in exchange mechanisms for fresh air going in and out of a house, but the heat gets conserved by countercurrent flow. And here you see this drawn out, this idea of optimal difference in the concentration gradient. This is concurrent flow, where you have the blood flowing in and the water. Here, this blood flowing in with the concurrent flow. This is not the way it is designed. It's in fact shows that it's not this way. It is countercurrent flow. If you look at the differences between the blood flowing around the water, past the water, of course they're divided. The blood is contained within the capillaries. Here you see that towards the end there would be no real difference and therefore no real exchange. But with a countercurrent flow, you get exchange over the entire length, because there's always, pretty much always a difference. Here it's the same. Quickly it becomes this very close. Here it is always, there's always some gradient difference. Okay, that's a countercurrent mechanism. Well, there are, in fact, in humans, the adaptation for breathing is that we are tidal breathers. We breathe into, and other vertebrates breathe this way also. We have our lungs internalized. Breathing is tidal. It's in and out of the same opening. It's actually less efficient than the gas exchange mechanism that fish use. Volume exchanged with one breath can vary considerably. You can inhale and exhale at rest. It's called tidal breathing. So you're inhaling and exhaling a tidal volume, but you have the capacity, and that's shown on the graph here, but you have the capacity to increase tremendously both in the amount inhaled and the amount expired or exhaled. You should, of course, realize that if you're breathing in and out of the same opening, the mouth, then you are inhaling. The inhaled air will always mix with some stale air, some air that has an increased concentration of CO2. So that's the reason why we say that the fish is more efficient, because it's a unidirectional countercurrent mechanism. The apparatus for measuring the different volumes of breathing is now simplified, what's called a spirometer. It's a very simplified mechanism where the nose is nose clips are used to block any flow of air out of the nose, and a structure that has a pressure transducer in it is linked up to a computer. Before, we used to use this kind of bizarre but effective apparatus, although a little bit cumbersome to work with, to get recordings. Well, as I said, in mammals you have the lungs. The gas exchange surface area in these internal lungs is huge, tremendously large. The diffusion path length from air to blood is very, very short. This photomicrograph down here shows tissue in the lungs and the alveoli. You have these alveolar structures that are very, very, very thin, surrounded by capillaries. So there's a very short diffusion path length, which is important. The other thing is, not only is there a large surface area from the millions of alveoli in the lungs, and not only is the length of diffusion path length very small or thin, but there's also a reduction, and this is an important feature, reduction of surface tension in the alveoli by secretion of surfactant from the type 2 epithelial cells, and that allows for much easier inflation. Otherwise, you'd get the alveoli collapsing and being held closed by surface tension of water. Water molecules are attracted to each other by hydrogen bonding, so that surface tension can be actually quite significant and would cause a difficulty in inhaling, opening them up, popping them open. This is actually the kind of problem that occurs in premature babies. They can be delivered quite young, and their lungs aren't fully developed, and they're not producing enough surfactant, so they cannot breathe often on their own, and they have to be assisted in breathing by positive pressure breathing, where the air is forced into their lungs to help pop open overcome the surface tension. Normally, surfactant does that, and so we can breathe by negative pressure breathing. Breathing, or pulmonary ventilation, has two phases. Inspiration, air is going in. Expiration, the gas is breathed out. It's a mechanical process that depends on volume changes in the thoracic cavity. When the volume changes, that causes the pressure to change based on the concept of Boyle's law, V1 P1 equals V2 P2. As you change the volume, increase the volume this way. I'm going to write it a different color. Just a second, sorry. As you increase the volume of the thoracic cavity, the pressure drops, and as a result, air flows in. So in the end, the two sides have to be equal in this equation. So air flows in and out, always down the pressure gradient. Airflow is occurring so that you get equalization of pressure between the lungs and the outside. When we consider pressure, there's one feature that's important and that you should know right away, is that the normal atmospheric pressure at sea level is 760 millimeters of mercury, and that's it's always there, it's always present, it's pressing on the chest, it's in the atmosphere around us. So we ignore that and we only talk about the difference above or below that. Okay? And so when we look at pressures, you can see that these are the kind of pressures we're talking about, and these pressures ignore the atmospheric pressure. So we're talking about pressure above in the alveolus, in the space of the lung, pressure above in the alveoli, above atmospheric, causes exhalation, and if the pressure is below atmospheric, then it causes inhalation, then air will flow in. The key muscles involved in inhalation at rest are the diaphragm and the external intercostal muscles. The word external is missing here, but that's important. External intercostal muscles. What they do when they contract is that they increase the volume that the lungs are, the space around the lungs. You see, this is this is what happens when the diaphragm contracts. It moves down because it's a shorter distance, so it has to move down. The chest wall rises, and you get a increased volume, and then therefore a decreased pressure, and you get inhalation. That's what's happening on that side of the diagram. When you relax, the diaphragm pops up. It goes into that dome-shaped. The thoracic cavity space gets smaller. The chest wall moves down, so the volume is smaller. Pressure rises, and you breathe out. So when you look at it, it says here other intercostal muscles contract. Those would be the internal intercostal muscles. So external intercostals are involved in inhalation, diaphragm and external intercostals. Mostly exhalation is just a relaxation of the muscles involved in inspiration. Other muscles get involved when you're doing very active exhalation, like when you're running and you're breathing out, you try to breathe out faster. Now I want to point out something to you, and that is that, remember, the lungs are surrounded by a membrane. On the lung surface of the lungs themselves is the membrane, the visceral pleura, and then on the wall of the cavity in the thorax there's the parietal pleura, and the space in between is the interpleural space, and there's fluid in there. Now fluid doesn't change in volume very much as opposed to gas, but it certainly can change in its pressure. And when you contract the inspiratory muscles, the inspiratory muscles for inhalation, which are the diaphragm and the external intercostal muscles, and you get a big increase in the volume, then the pressure drops. in that intrapleural space. And you can see the pressure dropping. The pressure in the intrapleural space, so the pleural cavity, is always greater than the intrapulmonary or intraalveolar. It can be called intrapulmonary or intraalveolar. It's always greater because with that drop in pressure, then it pulls the lungs. The lungs will also expand because they're pulled by that intrapleural pressure, that drop in intrapleural pressure. This pressure is always less than the alveolar pressure. If they were the same, then the lungs would not be able to be pushed up against the wall of the cavity, and they would collapse. So, in terms of inward forces acting to pull lungs away from the thoracic wall, forces that promote lung collapse, you have the elasticity of the lung, tending to pull them towards the smallest possible size, and you also have the surface tension of the fluid on the inner lining, on the inside space in the alveoli, also tending to pull them towards their smallest possible size. The opposing forces that keep the lungs enlarged is the elasticity of the chest wall itself, pulling the thorax outward to enlarge the lungs. Boyle's law, as I told you, P1V1 is equal P2V2. It really represents the initial and end conditions in terms of inhalation and exhalation quite nicely. Again, in terms of lung pressure relationships, you have that intrapleural pressure in the space between the two pleural membranes. That's intrapleural pressure. You also have intrapulmonary or intraalveolar, same thing, pressure, which is the pressure in the space inside the alveoli. And the difference between the two is called the transpulmonary pressure. This is what keeps the airways open. You should notice, as I said, I'm not sure, as I said, it's always less than, the intrapleural pressure is always lower than the intrapulmonary pressure, and therefore tends to pull that, the pulmonary pressure or intrapulmonary pressure, intraalveolar pressure, tends to keep, because it's greater than the intrapleural pressure, it tends to hold the lungs against the walls. If the two are equal, as I said before, they would collapse. Now this slide, what it shows are the events associated. We're going to go through the events associated with inspiration again. And that's what's happening on the left side here in these two graphs. What you have is the diaphragm and the external intercostal muscles contract, the ribcage rises, the diaphragm drops, the lungs, not the legs, the lungs get stretched out in volume, their volume increases, so the intrapulmonary volume increases, and the intrapulmonary pressure drops below 1 atmosphere, below the normal atmospheric pressure drops about 1 millimeter of mercury, and you see that here. That's the intrapulmonary pressure dropping down, and the air will flow into the lungs down the pressure gradient. Because it drops below atmospheric, then atmospheric is higher, and so flow is always down the gradient from higher pressure to lower pressure. At the point where you get the pressures being equal, which is of course right here, where that pressure line crosses the zero, because we've zeroed it against 760. 760 is the normal pressure of the atmosphere. So as soon as it hits that zero, then airflow stops, and the lungs you don't inhale anymore. When the intrapulmonary equals atmospheric, there's no more inhalation. Now expiration. The events here are shown, the events associated with that are again shown here. The muscles, the sequence of events, the changes in the shape, and the changes, anatomical changes there. Now for expiration, and that's shown on the left side here, the The inspiratory muscles relax, the rib cage descends or drops down, your chest is lowered, not as raised. The thoracic cavity volume will decrease and the elastic lungs will recoil. They were stretched before. Remember that the alveoli have elastic fibers around them. So when you inhaled, you were stretching those out. So now you'll get some recoil. The intrapulmonary volume will drop and therefore the pressure rises. As the intrapulmonary pressure rises above atmospheric, you'll get flow of air in. And you'll get flow of air out of the lungs to the atmosphere. So gas flows out. And it keeps doing that until it hits this point where pressure goes back to being equal, no difference between that and atmospheric. Now, I haven't talked too much about the pressure in here in that intrapleural space, but again, it's always below whatever the pulmonary pressure is, and that's what helps keep the lungs pressed up against the wall. And again, you can go through that, what we just talked about, what's happening to get expiration. Remember, expiration is really a passive process, whereas inhalation is the active process. Okay, so here you see again the transpulmonary pressure is the difference between intrapulmonary and intrapleural. And intrapulmonary is always greater than intrapleural all through that sequence of events in inspiration and expiration. Sometimes people get a tear in the visceral pleura that covers the lungs and into the lung tissue, and you get air suddenly going into that intrapleural space, boom, the lungs will collapse. Or if they have a wound from the outside through the thoracic wall into that intrapleural space, air will get in and lungs will collapse, because there has to be a difference in pressures. The intrapulmonary must be greater always than the intrapleural, and that's what helps hold the lungs up against the wall.